Welcome back. Uh, Hate allergies. Welcome back. We have density gradient centrifugation. This is an important technique for purifying proteins, certainly nucleic acids, and for organelles. Okay, this doesn't show that, but you could add organelles to your notes. So we've talked about differential, and I did mention there were three uh, main types of centrifugation. So there are two types of density gradient centrifugation used for different purposes. So those are zonal or rate zonal, usually with a sucrose, very viscous, dense gradient centrifugation, or isopycnic, isodensity, use that term maybe to understand it. And for DNA, we would use cesium chloride, and there's all other types of media, Percol, Fico, all sorts of different media that one uses depending on you're doing isopycnic or zonal. Okay, so go back, make sure you got differential centrifugation, and now we're going to get into zonal or isopycnic. But just remember, those are a type called density gradients. So there's actually a media that the proteins, the organelles, the samples are in, and then there's two different techniques shown here in blue to use to move down the line. So you can catch this later here. So we'll start off with rate zonal centrifugation. And so we have a cartoon here showing the beginning, the results of centrifugation here, and then a step that has to occur, and this is a subject going later, but it's fractionation. Okay, it's fractionate. So the sample is loaded on the top, and what we have in this is a sucrose gradient. A gradient's definition means that there's a low amount of material at one place, and then a high amount of material at another, or in this case, low density to high density. So we have low density in this region of the media and high density. So right away, you see that various organelles, other kinds of macromolecules will move through this media based on their density. And so we apply a centrifugal force, and we end up with fast sedimenting material, components, organelles, and slow. Okay, so so we have basically two bands here. Okay, two bands, and you're saying, how do we visualize these? We'll talk about that later, but there are ways. Sometimes there's just a dense haze. This is sucrose, which is fairly clear, and so it's kind of like looking through cellophane or a clear piece of plastic paper, and we can see there's material here and material there. Okay, and then there's a process known as fractionation, which then collects slices, okay, of this gradient. And so they're showing this fractionates here, and we have some areas that don't seem to have material, and then we hit this, I'm sorry, that's the slow, this is the fast. Okay, and then we have some other. So in this very simple cartoon, we have two fractions. So if this is, say, 10 mils, then, okay, 10, 20 mils, then these would represent 2 mil fractions. So we have to have a machine and a process to grab 2 mil slices at a time without shaking this up. Obviously, if you shook it up, if you dropped it on the floor, and by the way, this would usually have a top, but not always. Okay, and if you dropped it, well, it would leak out. Obviously, you'd be mad, but if you dropped it, you would shake these up, and you'd have to recentrifuge if you had any material left. So this is carefully removed from the centrifuge rotor and then applied in a fractionation machine and process to get sample areas. And then we have our two basic slices here. This is also known as velocity sedimentation. Materials driven by the speed of the centrifugation will sediment at where their density kind of leaves them. Now, time. Rate zonal cannot be done for too long, because given enough time, all these will pellet at the bottom and you haven't done anything. So there's a time issue here, okay? So you can't sediment for too long. Obviously, if you spin it for 5 minutes, this sample probably won't move, okay? If you spin it for 3 days, you just get a pellet. So we're talking empirical again, empirical testing. You may have to test to see under your conditions, your cells, whatever the disease, anything that's going on that's different in your hands, phases of the moon, whatever it is, you may have to do empirical studies to try to figure out. We will see later that companies will actually sell you density beads and in a little kit, and you can put these beads, and they have different colors, and each bead has a certain density, so you can actually practice with positive control with density beads, right? And you can see how that changes over time, and we'll see that. I've got a great slide. So when you see that slide, kind of feedback to this slide when we're discussing the time issue and the empirical ways that we have to deal with rate zonal. So I think I just blow up this cartoon here. Rate zonal, the sample is at the top, and then you have a gradient. And you're saying, well, how do you get this gradient? Two ways, three ways actually. Some companies will actually sell you a centrifuge container closed that already has a pre-made gradient. You can buy a machine called a gradient maker. You add a certain high density sucrose, and it will actually mix it with the buffer and put a low to high sucrose gradient formed here. You could do a layered where you layer different little cake slices that are very dense to low dense and kind of have a layered gradient. Okay, that's less sophisticated than this complete, more differential gradient. In other words, you have 1.05, 1.051, 2, 3, 4, 5, 6, 7, 6, 1, 6, 2, 6, 3. 6. So you see, you having a small slice, a little bit denser each time. Could be empirical. Okay, EP, empirical. I may have to test this, right? And of course, you need to confirm what it is you're looking for. Are you looking for these or these, or both, depends. Rate zonal also is given the name moving zone. Part of biology is to have multiple names for one kind of process to trick students. No, that's not it. Just different companies, individuals give different monikers to a process and it gets stuck in the literature. So in this cartoon, we have an initial position. Here's your sample with organelles and material. And then there's the, say, 4 slices. And they're trying to show you, see how the colors get dark? That reflects a density layers, light to heavy density. And so initially, and then with enough time and spin, in the centrifuge, you will get some separation of fractions here. It looks like one ends up in 1, this material ends up in fraction or area 2. Okay, preparation for the media, and it shows the lightest, or the, I guess it's going this way. Yeah, so it's, I wouldn't have done this way. I would, uh, this needs to be reversed because your sample's here and then you go down in density. This is kind of an opposite way to put that. But anyway, samples applied at the thin zone at the top. So rate zonal always has a sample at the top, and then you have some type of density viscous gradient from low to high density. Okay, so just kind of while you're describing that on the test, just, you know, think how you could describe that. And so under centrifugal force, the particles will start moving. And like I said, given enough time, they will pellet. So you can't, yeah, so insufficient time, incomplete. If you go over time, basically that means they all pellet. And again, based on size and density. So you end up getting separate zones here, and those will be able to be separated given enough time. So this may be using somebody's protocol, but remember that, you know, if it's a new cell or situation, somebody's protocol may not work for you. What do we say? Empirical. Okay, empirical studies here. Okay, so now we're moving to the second type, and it's called isopycnic, also known, to trick students, no, equilibrium, sedimentation, centrifugation. Goodness, isopycnic is shorter, right? So notice the first thing that's critical. So if you're writing about these two, this is what distinguishes them. Sample is mixed. So you take a steep gradient, that means 20 to 70% sucrose here. You mix the sample up and you apply centrifugal force. So here it is. And by the way, this is a comparison, so I may not have. This is the rate zonal, rate zonal here. Okay, we have the sucrose gradient and the samples on top. Isopycnic, the sample is mixed. Rate zonal, the sample is sitting right there. So sample is all through here, sample is right there. Okay, and you end up getting the low density components, high density. So these things move up or down in this gradient depending on their density. Whereas in rate zonal, they start at the top and move only to the sample doesn't start at the bottom, okay, and move up. It starts here. Whereas here, notice they're trying to show you, it's hard to tell, but notice There's starting to be a separation of the red with a little blue. So the red and blue evenly mixed. After some time, a lot of red here, a lot of blue here, and then given enough time, they band into tight areas because that's where they are equal to the density in that part of the gradient. So there's an equality here of sample density to the gradient at that point. So there it is right there. Mixed sample, isopicnic, and they go up or down depending. See, some red's moving up, right? So the red down here is moving up. The blue up here is moving down. And they're finding where they're sitting right on top the gradient where their density is. So they can only go to where. So time-wise, not no issue with time. Okay, because of the way this is working, these materials stop at the gradient and can't go further. Okay, this can keep going down. Okay, over time. Okay. So here it is, just showing that again, moving in and out. So iso picnic, you mix, and then material goes up or down depending on whether low, medium, or high density. Sample has to be evenly distributed throughout to begin with. So this is also known as a self-generating gradient. Since you mix everything, the gradient is not formed. The centrifugation actually forms the density gradient. The previous one, the rate zonal, the gradient's usually already made, and you just put the sample on top. Big difference. Okay, big difference. So here's another way to compare them. Okay, rate zonal, samples at the top, and you have a movement through the various layers. Isopicnic, the sample's mixed, the gradient's unformed, and then centrifugation, over time the gradient forms. Here the gradient's already formed. This is lower speed. You don't complete it, right? You have so different density, similar density, different molecular weights, cell organs, organelles, and nucleic acid. So time-wise, you do have a time limit. This pretty much long-term, high speed. The density is equal, iso, equal. Similar molecular weight, but different densities. Okay, you're separating. So similar densities, different molecular weight. Proteins, similar densities, but different in, so but proteins have about the same density, but they're different in molecular weight. So this is a better way to separate proteins. This is better for DNA, RNA, and organelles. So if you're describing the usage here, this tends to be for proteins, this tends to be for organelles. So I thought this was a good slide to kind of hammer some of these subtle parts. Can you see where students get confused or flip, and then with differential flipped in, and all of a sudden they're mixing their metaphors. All of a sudden it's not good. Okay, so this is a trap. You're warned about it. Whether you want to step in or not, that's your job. So rate zonal versus isopicnic or equilibrium. Okay, just another figure. I don't know where I stole this from, but yeah, moto photo. Anyway, just shows, and then our fun fractionation there at this point. And again, these materials may not have color metric, or they could. You could stain them. Okay, so here's a chart comparing the isopicnic. See right here, iso versus rate zonal. So take your notes. See, they say zone, but they should rate zonal, isopycnic. Okay, they mention isopycnic here. Rate zonal. The gradient is shallow, stabilized. The gradient is very steep, from like 20 to 70% sucrose. And so you're saying, why is that? You've got to have the maximum gradient density greater than anything in that mix. Whereas here, you don't have that. That's why there's a pellet. If you go too quick, is because the material you're spinning probably is more dense than the material it's going through. Whereas here, that's not the case. 70% sucrose is almost impenetrable. Okay. And in fact, a lot of people collect viruses from virally infected cells. They put them through an isopycnic centrifugation and put like 80 or 100% sucrose at the bottom, and all the viruses end up right at that thin layer at the most dense, and you could just remove the gradient one at a time till you get to that little virus layer, and then remove that, and you got almost pure crystalline virus here. Centrifugation is incomplete, short time, low speed. This is sediment to their equilibrium position because we have such a steep gradient. Very long time, very high speed. Okay. This one, of course, you can do this quicker. This is for organelles. This is more for protein work, and we'll talk about protein fractionation here. So here's our blood that if we separate out, so some of the earliest centrifugation for the clinical side was just separating your red blood cells, your leukocytes, and your plasma. And so for blood donors and things, you could look. If somebody had like leukemia, they would have a lot of white blood cells, and the leukemia buffy coat would be much bigger. So right away they could tell when they took your blood, I mean, if they couldn't figure out by the other symptoms, that you'd have a lot of white blood cells. Okay, it's normally 1%. Okay, when that gets 5, 6, 10, there's issues. Okay, so here's your centrifugation. Here's blood, and then here's your red blood cells. And notice, this is what I was discussing. You have such a dense material here. This very dense cannot go into this. It's dense, but not as dense as that. So it kind of catches everything here, the dense. Okay, so viruses would be in this area, that buffer zone there. And this is just another cartoon. So before and after centrifugation, quick centrifugation. And so you can measure somebody's hematocrit, the amount of red blood cells, just with an estimation of how much you see. How is this layer here? How thick is it? Okay, so you can get some good clinical information, just a real quick spin, just a look-see spin. And then here's just the centrifugation model we talked about. So to remind you, differential centrifugation can get us organelles. We can do the various pellets and soups, increasing G-forces and time. We can get mitochondria, lysosomes, microsomes, and then the soluble proteins here. You have to have broken the cells up tenderly, neatly with protease inhibitors. Okay. And so you can break up into these fractions. So you pellet into pellet and soup, and you keep doing that. Whereas gradient, let me drink something here. Okay, so separate into homogeneous according to their densities, rate zonal and isopycnic. The rate zonal is on a preformed density gradient. Like I said, you can buy a lot of these already pre-made, or you can make them yourself. Isopycnic, you add enough material to end up with a steep gradient, but you mix your sample in, and That's longer with a lot more speed. Rate zonal is quicker here. Rate zonal will be organelles, and isopycnic tends to be proteins. Differential tends to be organelles. So we have two methods here. Uh, Differential is quick, dirty, and that may be all we need from initial. Rate zonal is a little bit more cleaner. Here's cartoons showing the rate zonal separation, and eventually uh, in isopycnic that they will form the two areas here over time. And rate zonal, you start getting separation here, but if, like I said, eventually they could pellet them. And so here's buoyant density of things like the viruses for us. This is the dense Svedberg unit, so this is uh, lighter, and this is the more dense here. Okay, so you can start separating. There's a gradient called Percol, which is artificial, and a lot of lab just cheap to get sucrose. So but this may be a little bit more. Well, you can see that notice mitochondria and lysosomes, how they're close together with Percol, but sucrose, they're a lot more separate there. So you can get, depending on what you're looking for, some differential separation depending on the gradient you make here. This is our lipids, the LDL, the HDL, the good lipid, the bad lipid. So when they take a sample and do a lipid test, they could spin it. And so what happens is you can have a laser that scans through the sample and measures the intensity. So there is the V, very light LDL. This is the LDL, and this is the high density lipid. You see this is a lot more spread out. Notice that spread. This is a lot tighter, and this is the tightest. So this is the result, and this is the trace, and notice the density is low to high. Okay, and so we can actually, this is triglyceride, little peak in here as well. So we can look at your lipid profile if we had enough time and get exactly how much you have. You can integrate, if you remember your calculus, maybe you hadn't had, you can actually integrate these peaks and figure out exactly how much of total each one of these fractions represents, so you can get a percentage of high versus the good versus the bad cholesterol. Okay, and there's all sorts of different materials that you can get, something called NicoPrep, there's Percols, and they were just, oh, that was good. Maybe it's about time for me to shut up. Oh, it won't go back. Anyway, there's different media here, and just remind you that rate zonal is probably what we'll be using for organelles here, and just remind you about a preformed gradient, and you separate the material here. So, and here's the colored beads that you can order from, here's a company, Amisham. And so in 15 minutes, 30 minutes, 60 minutes, and if you went 90 minutes, notice everybody's at the bottom. So each area is a certain density. And since we've done this enough, informatically, we know what organelles go here with this Percol gradient with, we know exactly how much Percol, we know exactly what these beads and how they distribute. So running a positive control tells you the time you probably need to do on your rate zonal. Okay, so you're asking, how do we visualize this? They have colors. You can take a picture of this, and then you can figure out exactly what these densities are, and you can plot on a graph paper the densities and see the differential here. So again, too much time and everybody's pelleted, and you've lost a lot at that point. Not a lot of separation at that point. Okay, just going to introduce fraction collectors. Uh, it's hard to see. There's a flask with your homogenate, with your centrifuge bands, with your material. There's some tubing that goes into here. It's got to be sterile. You don't want just fungal and infested. And that tubing goes up and is pumped. So there's a vacuum and material is pumped up from your sample here. So this is a sample. Whoa. Sample here. Sample. Hey. Shh. And pumped. Shh. There's nothing there to eat. Hey. So sample up through the tube, and each one of these is a test tube, and that's the fraction. So when we talked about fractions, that's a fraction collector. Okay. And so here's another fraction collector. So each tube represents a different slice of the fraction. Here's another one as well. So this is a robotic arm, and it moves down the bingo card. If you think of this as a bingo card, what happens if the robot breaks? Then all your fractions end up in one tube. You know, you go down to, Corky's barbecue here, and you think this is working, and everything just overflowed. You think that's happened to me? Yeah. So you pretty much have to do this, and a lot of times this is done in a walk-in cold room, 4 degrees. So you have to wear your parka and you jump in and out to make sure this thing is moving. And you want your, remember we added that protease inhibitors, so they should still be working as well. Okay. So this is after the centrifugation, not before. This is after, and we end up with fractions. Now, how do we test these fractions here to see what's in each one of them? That leads us to spectrophotometry and protein analysis that we will pick up. I know you're happy that I'm done. We will pick up in our next lecture. So I'll start here with the spectrophotometer, and we'll go from there on how we test each fraction. Okay, come back. We'll have fun. See ya.